Develogin was founded roughly 10 years ago by Peter Gruss, a fairly well-known mouse geneticist, on the premises to use functional genomics to identify really novel targets, novel pathways to address diabetes and potentially develop therapies that are really disease-modifying, potentially even can reverse the progression of the disease. I operate the slides here myself. Okay, so before I go into the depths of the programs that I want to talk about, I briefly want to reiterate and reinforce some of the notions that were done here before from my colleagues. Type 2 diabetes really is a complex disease, and it's not simply defined by elevated blood glucose levels or hyperglycemia. Not at all. There are various other aspects to diabetes which are shown here. So it's in particular characterized by insulin resistance, an important driver of the disease, elevated plasma lipids, but also in particular loss of functional beta cell mass, as well as weight gain. And I think what's important to point out here that hyperglycemia or elevated blood glucose levels are really a fairly late stage of the disease already. So that by the time hyperglycemia is observed, this is already very far into the disease progression, whereas other components of the disease, such as insulin resistance, elevated plasma lipids, loss of beta cell function, happen much earlier in the disease progression, as you can see here. In terms of insulin sensitivity, for example, it drops already fairly early. Regards to circulating free fatty acids go up fairly early, way before you can see an elevation in plasma glucose levels. What's interesting to note is that if you're looking at the available therapies currently on the market and also in development, most of them only address a couple of these aspects of disease, in particular hyperglycemia usually, and in some cases also the lipid profile. But they do not address very important other function of the disease. I'll come to that here. So as you can see on this particular slide is that metformin, for example, a drug that's widely used for the treatment of diabetes, has insulin sensitizing activity, particularly in the liver, suppressing hepatic glucose production, and thereby lowering blood glucose levels, and has a slight positive effect on body weight as well, which is unfortunately not sustainable. But it has also clean safety profiles. But it has deficiencies in the area of the lipid profile as well as the beta cell mass, which are not addressed by metformin. Similarly, for sulfonylureas, which are mainly driving the insulin secretion of the beta cells, but don't really do much else for a diabetic patient. The TZDs or glitazones are a very effective drug class in terms of addressing insulin sensitivity and glucose levels, blood glucose levels as well as lipid levels. But unfortunately, they lead to weight gain and have other safety concerns associated with them. And more recent developments in the area of diabetes are DPP-4 inhibitors, so-called incretin enhancers, which also are sort of insulin secretagogues that are glucose dependent and help to lower blood glucose levels, but once again are deficient in other areas. And that's the same is true for the SGLT2 inhibitors that are currently in late stages of clinical development. What we have tried to do at Develogin is essentially try really to identify completely novel targets based on functional genomic screens in model organisms. And we've carried out here a genome-wide screen in a model organism such as the Drosophila genome, which is a widely accepted genetic organism which allows quick or fairly rapid screening of a genome. In this screen, we have looked for clinically relevant readouts such as triglyceride levels, glycogen levels, etc., and have identified a very large number of potential target genes, which we have then subjected to a very rigorous selection process. In this process, we have gone right in the way into the human genome to identify the respective human counterparts of the genes we identified in the model organism, then carried out extensive expression profiling to see if the genes were expressed in the disease-relevant tissues, metabolically relevant tissues, and then have carried out quite extensive in vitro and in vivo functional studies to really tie down in which processes these genes are involved before we went on to the drug discovery process. This is just to give you an idea what sort of came out of this screen, and at the very top of the list is a target that we are particularly fond of, identified at the Village. It's what we call DG70. It's a novel kinase that suppresses insulin signaling and thereby leads to insulin resistance type of syndrome. It's located in the insulin signaling transduction cascade, and we have worked quite hard to characterize and validate this target, and have also carried out drug discovery to come up now with compounds that can inhibit this target, and which are really now an emerging class in our minds of new class of insulin sensitizers. What we've done is we've extensively characterized the genes and validated this particular target, so we know it's expressed in metabolically relevant tissues. It has high levels of this gene lead to the insulin resistance phenotype that carries all the detrimental effects that are associated with diabetes and obesity. We have also been able to show that in vitro and in vivo functional studies, if you knock down the function of this gene, you get increased insulin sensitivity. Now, in the drug discovery process, we have been able to identify very highly potent compounds that inhibit this particular gene very specifically, and have carried out a number of in vivo pharmacology studies which have always led to a very consistent phenotype of improved insulin sensitivity, improved glucose tolerance, normalization of blood glucose and lipid profiles, as well as the preservation of beta cell mass and a decrease in weight gain, which is really unique in this field. So essentially, with DG7 inhibitors, we are able to really target the complete spectrum of phenotypes associated with type 2 diabetes obesity. This is just to give you sort of a glimpse at the characterization of these compounds. In one particular model, this is a ZDF rat model, where we here looking at glucose levels in vehicle-treated animals that are shown in blue, and rosiglitazone-treated animals in pink. And then we have the DG7 inhibitors in 2 doses here in green and light green and dark green. And as you can see, DG7 inhibitors are able to lower, improve blood glucose levels to the same extent as rosiglitazone, which is a very potent agent in this regard. They are also able to have a beneficial effect on body weight gain, in contrast to the TZDs, which is a very important aspect. If you look at the lipid profile, you see a lipid profile that is also, once again, very similar to the efficacy of rosiglitazone in this case. And now, very important, and what really sets this approach apart is that in contrast to other approaches in the field, we have a very robust effect on the preservation of beta cell mass, really maintaining the endogenous insulin secretion capacity of this organism, in this case mice, and thereby able to contain hyperglycemia very nicely. So if you look at our approach compared to other approaches in the field that are currently on the market or in late stages of clinical development, you can see that DG7 inhibitors are really able to tick all the boxes, all the important boxes that are the core problems of type 2 diabetes. By really targeting the underlying problem of the disease, insulin resistance, we are not only able to improve blood glucose levels, but we are also able to improve or normalize lipid profile, preserve the beta cell mass, and lead to a slight decrease in weight gain. Since the target itself is not associated with any safety profile, with any safety issues, we do not expect to encounter any safety issues in the long term that are in any way target related, and as long as we devise highly selective inhibitors, this should be free of any side effects.